Good day everyone. Welcome to the course on next generation sequencing technologies, data analysis and applications. In the last class, we introduced different types of epigenetic modifications, namely DNA methylation, histone modifications and chromatin remodeling. In this class and the remaining classes, we will be focusing on how we can apply next generation sequencing technology methods to detect these epigenetic modifications. So, in this class we will be talking about DNA methylations and how we can apply next generation sequencing technologies to detect genome wide DNA methylation patterns. These are the keywords for this class, methylation and bisulfite. So, from the last class we introduced DNA methylation. So, these are modifications that are directly added on the DNA base. The modification here is methyl group, and this modification appears mostly on A or C base. So, another point is that these are not just methyl groups, they could also be other groups, for example, hydroxymethyl or carboxyl groups. We will see in a moment. I will show you some of these modifications, and we will see there are a variety of other groups that can also be added, although these are rare. The most common one is the methyl modification on A and C. So, let us talk about the methylation on adenine base. Okay. So, the A base looks like this on the left. So, you see the A base and we have the 6 methyl adenine or 6 MA in short. Okay. So, you can see this methyl group added here, okay, on this nitrogen. Similarly, we have methylation on cytosine, and again I have drawn these modifications for you. So, on the left you have the cytosine, then in the middle you have the 5 methyl cytosine. This is the most common modification of cytosine that we see. Okay. So, here is the methyl group, and you also see 5 hydroxymethyl cytosine. Okay. So, here is the CH2OH. So, you can see we also see this modification quite commonly in the genomes. And again, these two types of modifications on cytosine, they are associated with different outcomes and they affect gene expression. So, 5 hydroxymethyl cytosine will denote by 5-HMC. There are other modifications on cytosine base as well, but these are rare. Okay. So, compared to the 5-MC or 5-HMC, these two modifications are quite rare in the genome. So, we will not be focusing on them today, but at least I should mention what are these modifications. So, one is formyl cytosine, okay, or 5-FC, you can see this CHO group, and then on the right you have the 5-carboxyl cytosine, or we refer to as 5-CAC, and you see the COOH group. Okay. So, these two modifications are rare. So, we will not be talking about detecting these type of modifications, we will be talking about detecting the other ones, right. So, these will be 3 modifications now. Okay. So, we have 6 MA, that is modification on A, we have 5 MC and 5 HMC. Okay. So, let us first focus on this cytosine base and how we can identify methylation on cytosine base. Okay. So, this has been an area of interest because these modifications were quite common, okay, and they appeared in the CpG islands of the promoters in the mammalian genomes. Okay. So, presence of these modifications, they actually had silencing effect on the gene. So, that is why researchers wanted to study these modifications in the CpG islands mostly. Okay. So, what we will do is we will talk about some of the traditional methods that were employed to actually identify this methylation, and this was before NGS era, and this will actually give you some background, right, some of the strategies that can be applied. And what you will see is that after the advent of NGS, we can actually extend some of these techniques for identifying these methylation patterns on cytosine base. Okay. So, these methods already existed and they simply been extended now with application of NGS, and then we will talk about a method that actually applies NGS liberally, and then we can study these genome wide patterns on cytosine basis. Okay. So, let us start with the traditional methods. Okay. So, these methods can be classified under different classes. I will be just touching upon them very briefly, and I will just mention the principle. Okay, I will not have time to go into the details. If you are interested, of course, I will share all the references so you can go through them. So, the first type of method relied on something called restriction digestion based detection. So, I am sure you are aware of restriction enzymes. So, these methods use those restriction enzymes to identify the methylation patterns. In the next slide, I will actually discuss this in a bit more detail. Then we also had PCR amplification based method for detection of this methylation pattern. We also had affinity based methods, and then of course, we had Sanger sequencing. Okay. So, Sanger sequencing could allow us to detect some of these regions, right, showing changes or showing this methylation on cytosine. And then there was microarray techniques that actually allowed some level of high throughput. Okay. So, all the methods are dear methods, they were mostly low throughput in nature, but then with appearance of microarray techniques, this actually allowed some high throughput analysis, and now this has been extended with next generation sequencing technologies. Okay. So, I will discuss this restriction digestion based detection method very briefly. So, the principle is actually very simple. Okay. So, we use restriction enzymes that can digest DNA, and they are also sensitive to methylation. Okay. So, if you have an enzyme that actually can detect this or cuts around this CG base or CPG islands, okay, and if this enzyme is sensitive to methylation, so if a methylation is present, the enzyme will not cut, okay, but if there is no methylation, the enzyme will cut, right. So, by looking at the restriction pattern, you can differentiate, right, between unmethylated and methylated bases. Okay. So, that is the idea, and there are some examples of such enzymes where you see the restriction sites or the recognition sites for this restriction enzymes, and then the CPG island, right, or here you have this CPG methylation, right. So, you have the CG bases. So, any methylation on C would block activity of these enzymes. Okay. So, using these enzymes you can then differentiate, okay, which DNA fragment has methylation and which DNA fragment does not. Okay. So, there is a specific method that actually employed this technique, is called restriction landmark genome scanning, or RLGS. So, what happens is that these restriction sites act as landmarks in the genome, right. They are scattered around the genome at different locations, and they act as landmarks, and which you can use to identify, okay, whether there is methylation or not. So, this method actually applied this restriction digestion following the same principle that I just mentioned, and then it separated these fragments using 2D gel electrophoresis. Okay. So, very briefly, what will happen if you have methylation and if you do not, the pattern on 2D gel electrophoresis will be different, right. You can then separate out fragments, and then from that you could deduce, okay, where you have the methylation and where you do not. Okay. But again, you probably realize, right, because genomes are big, you cannot do this for a huge number of fragments, right. So, this means there would be limited throughput, right. So, you can do only for certain specific region or of certain size. Okay. So, this actually limited the throughput. Then we had the PCR amplification based detection methods, and very briefly we had something called methylation sensitive arbitrarily primed PCR. I will again mention the principle very briefly. Okay. So, here is an example, right. So, you have this DNA fragment, same DNA fragment, okay, and imagine this is the site of methylation, or C, right. This is the C base which can be methylated. Okay. So, in one sample this site is not methylated, there is no modification, and on this sample on the right you have this methyl group, you can see this methyl group, right. So, I have added in cartoon for this. Okay. Now, what will happen? So, we want to amplify this fragment, right, using these two primers on the end, right. So, you have these primers, and then we amplify, and both of them will get amplified, right. So, you will get a PCR product on the gel. So, in under normal circumstances, the methylation would not cause any problem, right, for PCR amplification. Now, what this method did, it actually applied a restriction enzyme that will actually recognize this site here around this methylation site, and it will cut if there is no methylation. Okay. So, again an enzyme that we just talked about, right, something like that, which will recognize this site and will be sensitive to methylation. So, what will happen is that on the left side this fragment will be digested, right. So, you will get 2 fragments from here. Okay. So, this will be digested, you will get 2 fragments, and then once you try to amplify you will not get any PCR product. On the other hand, on the right side, because there is methylation, the restriction enzyme will not work, the fragment will remain intact and you will get a PCR product, right. So, based on this principle you can then say, okay, which site is methylated. So, any region around the methylated site, if it is still amplified after the treatment with restriction enzyme, there that would mean there is a methylation, right. So, by this method you can then identify some of the sites where there is methylation. But again you can see, right, this will be limited in application or in throughput, because again if you want to do this genome wide you need these primers, right. So, again these primers are specific to specific regions, and you cannot have like 100,000 primers or millions of primers designed to do this. Okay. So, you can do this for targeted sites. If you are interested in certain genomic regions or certain promoters, you can do this. There is another method, I will not go into details. So, it is called amplification of intermethylated sites or IMS. So, it actually applies 2 sets of restriction enzyme digestion. So, one is the first enzyme is sensitive to methylation, the second one is insensitive to methylation, but they recognize same sequence set, right. And then finally, there is some sort of ligation and amplification, and what it will do finally, it will actually sequence or fragments that reside between 2 methylated sites. Okay. So, again you can imagine, right, this will identify only limited regions, and even with next generation sequencing you will not be able to identify sites that sit between 2 unmethylated C bases. Okay. Now, moving on to affinity based detection of cytosine methylation. So, the one method that we will talk about is called MEDIP or methylated DNA immunoprecipitation, and what it does is that it can detect 5-MC and 5-HMC modifications. So, it relies on affinity binding, right. So, you have an antibody that will detect this methylation and it will immunoprecipitate the fragments that carry this methyl group or the antibody. Okay, so, here is a cartoon for the process. Okay. So, here you have these DNA fragments, some of them carry these methylations, right. So, here you can see 1, 2, 3 etc., and some fragments do not have any methylation. Okay. So, in the next step what we do is we actually apply an antibody or mix with an antibody that will recognize these methylations, okay, that recognize this cytosine methylation, and they will specifically bind to DNA molecules that carry this methylation. Okay. So, you have generated these DNA fragments, some fragments will have methylation and some fragments will not have methylation. So, by applying this antibody we will have this antibody binding against this affiliation. So, this antibody has affinity towards this methylation, right. So, the specific antibody will then separate out DNA fragments that carry methylation, and the rest of the fragments will be washed away, and will just identify the specific fragments that bound to antibody. Okay. Now, we can identify these fragments using PCR, right. So, can combine with PCR and identify these fragments is called MEDIP PCR, right. So, again if you want to apply PCR methods you need primers. So, here you are testing against specific regions, right. So, you are kind of making a guess, right, and designing primers and see whether you are getting PCR amplification or not, right, and you can do this only for handful of regions or handful of promoter sequences. You can also combine MEDIP with microarray, right, this increases throughput, and finally, now you can apply this method with NGS and is called MEDIP-seq. Okay. So, here the main concern is the antibody, and the drawbacks are firstly the low resolution, right. So, we do not know the exact base where this modification is, right, because the antibody will bind a fragment, and in a fragment carrying methylation you can have multiple C bases, right, and you do not know which base actually was methylated, and for that reason the antibody actually detected that fragment, right. So, that is something you cannot know using this method. So, you have low resolution, you will know, okay, within let us say 50 base pair we have this methylation. It's also heavily dependent on antibody, right. So, if you have ever worked with antibody, you will see this is where the biggest challenge will be, right, getting a good quality and specific antibody raised against methylation. Okay. So, this process is heavily dependent on antibody, and the results will be influenced by that, and which means you also need to do control experiments, right. So, at the end of immunoprecipitation, you will see enrichment for certain regions, and this enrichment will be done with respect to the control experiment, right. So, we will have to compare against control experiments, and then you can identify the specific region. So, you can see the whole design is quite complex, right. So, you need to have this antibody, you need to have this immunoprecipitation, then you have to also have control experiments, then you have to sequence both and then compare them to identify regions. Even after all this, you will not get very high resolution. There is another method very similar, it is again dependent on affinity, but instead of antibody it utilizes a protein domain, it is called the MBD domain of methyl CpG binding protein 2. This is a protein that exists in cells, and this actually recognizes this methyl CpG group, right. So, on C base this methyl group is bound by this MBD domain of this protein. So, you can utilize this MBD domain to actually specifically take or pull down, right, this 5-MC, 5-HMC bases, okay, and you can do an affinity purification very similar to what we discussed just now, and then you can take those fragments and purify and then subject to sequencing, and you can identify those. Again, the drawbacks are again very similar, right. So, you have limited resolution, and again you have to run through a control experiment. Now, moving on to the technique that actually revolutionized this field of study of methylation on C bases is called bisulfite sequencing. Okay. So, this is a method that was described by Prabha et al. In 1992, that is long time before NGS actually came into picture. So, what happened is that this method could detect methylated cytosine and differentiate from the unmethylated ones. Okay. And what happens here is that you have bisulfite treatment of DNA followed by Sanger sequencing. So, we will elaborate this a bit more, and you will understand the principle in detail. Okay. So, the first step in this process is the bisulfite treatment. Okay. So, the DNA is treated with sodium bisulfite. So, what actually happens is that the cytosine, okay, that is shown in picture here, right. So, on the left this is cytosine. There are multiple steps, of course, I am not going to go into those steps. So, after these steps, what you get is the uracil. Okay. So, cytosine is converted to uracil by the bisulfite treatment. Okay. But so, if you see this, right. So, once you have this uracil, so if you do PCR on this DNA fragment and then sequence, this U will be sequenced as T. Okay. So, you will see you will observe T in the sequence data instead of the cytosine. Okay. So, keeping this in principle in mind, so one of the peculiarities that that is associated is that this will only happen if the C is unmethylated. Okay. So, this conversion happens only if the C is unmethylated. Okay. So, if the C is methylated or hydroxymethylated, this conversion will not happen. Okay. So, methylated cytosine remains the same and is sequenced as C. So, in the sequence data, you will observe that some bases remain as C even after bisulfite treatment, and some bases are converted to T. Okay. So, now based on this you can now say, okay, which base is cytosine, right, C or normal C, and which bases are 5-MC. Okay. So, you can identify those bases cytosines that actually carry this methylation. So, let us take this example of DNA sequence, so that you understand the concept better. So, here you have this fragment where you have two methylated cytosines which are highlighted here, right. So, you can see these methylations, and then you have two C's that are unmethylated. Okay. So, here are the two C's that are unmethylated, and then you have the two C's that are methylated. So, in the first step, when you do the bisulfite treatment, what will happen is that the unmethylated C's will be converted to U, right. So, I have highlighted them in red color. So, you can see these C's are converted to U, whereas the C's that contain methylation, they will remain as C. Now, if you then do PCR amplification, what you will get is that these Cs that converted to U, they will be sequenced as Ts. Okay. So, again they are highlighted in red color here. So, they will be sequenced as Ts, and the Cs that were methylated, they will be sequenced as Cs. Okay. So, based on this difference, you can now identify the bases, the cytosine bases that are methylated. Okay. So, what we do is then we have to sequence before bisulfite treatment and sequence after bisulfite treatment, and then we can simply compare, right, and then identify, okay, which are the methylated cytosines and which are unmethylated. Okay, so there are other methods that are based on bisulfite treatment, something called methylation sensitive single strand conformation analysis. I will not go into that or any of these methods. I will just simply mention them. You can look up if you are interested. There is something called high resolution melting analysis. These are methods that do not use next generation sequencing. You have methylation specific PCRs or MSPs. You have array based methods, Infinium from Illumina. So, now we can extend this bisulfite sequencing with NGS. Okay. So, remember bisulfite sequencing was developed a long time before NGS was in the picture. So, researchers used mostly Sanger sequencing, but now with the appearance of NGS, we can now apply this method to the whole genome. Okay. So, the process is very simple, right. So, now we treat the whole genome with bisulfite, and then we do the whole genome bisulfite sequencing. Okay. So, here is the paper which actually applied this method, and we can get this cytosine methylation at single base resolution. Okay. So, this was done for the human genome, and we could get this data for the whole genome. Okay. So, here is a very simple method, right, again without any complications of immunoprecipitation or any other method. Here you have simple treatment, and then you go and do the sequencing like simple genome sequencing, and you get the data. Okay. So, one of the limitations or one of the drawbacks of whole genome bisulfite sequencing is that when we are looking at the cytosine methylations, these are probably limited to only very small regions across the genome, right. So, there are only certain places where these methylation can happen, right. So, especially the CPG islands, for example, in human genome or in mammalian genome. Now, what we do in whole genome bisulfite sequencing, we are sequencing the full genome to search for these specific regions or look at these specific regions. What if we could reduce the amount of sequencing and actually only sequence the regions where the bisulfite sequencing or where these methylations could happen. Okay. And this is precisely this method suggested. It is called the reduced representation bisulfite sequencing or RRBSSEQ. Okay. So, in this method what happens is that so this is the paper which actually suggested this method, and this is actually a targeted bisulfite sequencing. Okay. So, what happens is that in this method we first digest the genomic DNA with restriction enzymes. Okay. So, and then generate these fragments. Now, we can use certain strategy to actually make your life easy. For example, you can target the CPG sites with enzymes that are insensitive to methylation pattern. So, what will happen is that now if you are targeting in and around CPG sites, you can then take those fragments, right, and then sequence those fragments, and immediately at the end of these fragments you will have these methylation patterns. Okay. So, you can imagine, right, from the whole genome you create these fragments whose ends actually contain mostly CPG islands. Okay. And these are the regions we are interested in at looking for methylation patterns. Okay. So, once you create this fragment and you sequence from the ends, you will get the sequence data. So, you do not need to actually sequence the full genome where you do not actually are not interested in studying or looking at this cytosine methylation. And once you have done this fragmentation, you can then add this adapter. One thing you need to remember, this adapter has to contain methylated Cs. So, if you have C base in your adapter, this would have to be methylated because of the next bisulfite treatment. Okay. Unless if your adapter does not contain methylated C, they will be converted to T, and then of course, this will cause problem for sequencing reactions, because we use the adapter sequence for primer binding, right, in Illumina. So, we do the bisulfite treatment, then we do the sequencing, and finally, we get the data from which we can analyze the methylation patterns. Now, one of the questions that we have not answered is that how can we distinguish 5-HMC from 5-MC? Okay, so we have seen we can now identify 5-MC, 5-HMC from C, okay, unmethylated C. How can we distinguish between these 5-HMC from 5-MC? Okay, so this has been seen that there is some importance of 5-HMC. So, in many cases 5-HMC could be a good marker of certain diseases, etc. So, we want to identify these 5-HMC modifications. Okay. So, as I mentioned that 5-HMC and 5-MC, they are not converted to uracil by sodium bisulfite. So, they will appear as C in the sequencing data. Now, there has been a method that actually was proposed in 2012, is called Tate assisted bisulfite sequencing or TAPSEC. Okay. So, this method can distinguish between 5-hydroxymethylcytosine and the 5-MC, 5-methyl cytosine. Okay. So, this is how this method works. Okay. So, again imagine this cartoon where you have different sites or different C's that are highlighted in orange, and they can be unmethylated, they can be methylated, and they can be hydroxymethylated. Okay. So, I have denoted them by no methylation. You can see there is no mark on them. Then you have 5-MC. So, this is methylation, and then you have 5-HMC. Okay. So, these are hydroxymethylated. Okay. So, what we want to do is we want to distinguish between this HMC and MC. Okay. And of course, the unmethylated C's also are there. So, in the first step what we do is we do an enzyme treatment, is called beta glucosyl transferase. So, what it does is it actually converts this 5-HMC modifications to 5-GMC modifications. Okay. We will see this is some sort of protection for the next step. Okay. So, in the next step there is Tate oxidation using an enzyme. Okay. So, it actually converts these 5-MC groups to 5-CSE. You can see this, right. So, here is these 5-CSE groups. So, they have been converted from 5-MC. So, this GMC protection actually is important, otherwise these HMC groups will also be converted to CSE. Okay. So, GMC actually protects from Tate oxidation. So, once you have this 5-CSE, now you can actually apply bisulfite treatment, and they will actually be converted to uracil, and they will be sequenced as Ts. Okay. So, what happens at the end is that these 5-HMC residues or bases, they are sequenced as Cs, and rest of them are sequenced as Ts. Okay. Now, with this data, what you want to do now is you want to have this 3-way comparison, right. So, why do you need to do this 3-way comparison? Because you want to distinguish between unmethylated cytosine, 5-MC and 5-HMC. Okay. So, for that what we do a 3-way comparison, right. So, we have to sequence before bisulfite treatment. So, we get the original sequence, then we get sequencing after bisulfite treatment. So, C bases that are converted to T, these are unmethylated cytosine. So, we identify them. Bases that remain as C after bisulfite treatment, they are 5-MC or 5-HMC. Okay. So, we cannot distinguish just after bisulfite treatment between 5-MC and 5-HMC. And now, we do the TAPSEC, and bases that are sequenced as C and which remained as C after bisulfite treatment, they are 5-HMC. Right. So, so using these 3 sequencing methods, right. So, you can actually determine this unmethylated C, 5-MC and 5-HMC. Okay. So, there is another method again looking at slightly different combination, is called oxidative bisulfite sequencing or OXBSSEC. It can also distinguish between 5-MC and 5-HMC. Okay. So, this actually relies on selective oxidation of 5-HMC to 5-FC followed by bisulfite treatment. Okay. So, C and 5-HMC, these are now sequenced as T, okay, and whereas the 5-MC, those bases are sequenced as C. Okay. Again, looking at these comparisons, you can derive which are unmethylated, which bases are 5-MC and which bases are 5-HMC. Okay. So, again this we compare with conventional bisulfite sequencing as I just mentioned. Okay. So, here are the references for this class, and to summarize, we talked about traditional methods for detecting cytosine methylation. We talked about PCR based detection methods, affinity based detection methods, and restriction digestion based detection methods, but they were all of either low resolution or they had low throughput or limited throughput. So, which we could not apply that across the whole genome. Microarray methods actually allowed some sort of high throughput measurements, but then again the resolution was concerned for many of these methods. So, bisulfite sequencing actually allows genome-wide detection of cytosine methylation at single base resolution, and we also talked about TAP-seq and OXPS-seq. These allow us to distinguish between 5-MC and 5-HMC bases, as we have just seen. And finally, we need to do 3-way comparison for resolving C, 5-MC and 5-HMC bases. Thank you very much.